Hello, I am Sai Chandana Koganti, and today I am excited to share my research project, which is experimental survey of image segmentation models on hematoxylin and eosin-stained images. I would like to start by thanking my professor for his support throughout this project. The main goal of this research is to find the most effective deep learning models for identifying and segmenting the nuclei, which are complex medical images that are stained with hematoxylin and eosin, also known as H&E staining. For this research, I explored five popular instance segmentation models, which are like mass-CNN, Detectron2, YOLO-V8, YOLO-V9, and HoverNet. Along with these models, I also explored two pre-processing techniques and widely used datasets like PanNuke and NuCLS. So yeah, this is the data pre-processing techniques which I have used. Before discussing about the models, I want to explain you about the pre-processing methods which I have applied to improve the image quality. Color normalization will reduces the staining variations in H&E stained images. So the techniques like color deconvolution and histogram matching were used to adjust the images. And the data augmentation step will help to make the dataset more diverse. I mean, by applying the transformations like rotations, flips, and also like scaling. So these variations makes the model more adaptable. By applying these pre-processing techniques, we were able to boost both the accuracy and consistency among the segmentation models. Now, let's look at the models which I have evaluated. This is the mass carcinoid model. This is the two-stage model. I mean, which is having two stages that uses a ResNet 101 backbone for a deep feature extraction. Next, it has a RPN, which is nothing but region proposal network. It will detect the objects, and region of interest will ensures the accurate alignment of the segmented regions. This is about mass-car CNN. And next, this is Detectron 2, which is a framework based on mass-car CNN. But it uses a ResNet 50 backbone with the feature pyramid network for a better detection. For better detection across the objects. Next, YOLO V8. So this is known for its speed. This single stage model predicts both bounding boxes and also the segmentation mask. I mean, in one pass, it is able to predict both bounding boxes and segmentation mask. Next, the YOLO family, but other version, which is YOLO V9. This model introduces the enhancements like PGI, programmable gradient information, and a reversible function to handle more information. And then, last but not the least, which is HoverNet. This model is specialized for dense and also the complex histopathology images. It has a dual branch design for both segmentation and nuclear classification. Now it's time to look at the datasets which I have used, PanNuke and NuCLS. So as I have previously said that NuCLS dataset has over 220,000 annotated nuclei from breast cancer cells. So it contains high resolution WSIs, whole slide images, which are split into small patches. So each patch has labeled nuclei and consists of 13 different types of cell types. So nucleus dataset is nothing but which is having 13 cell types like tumor, fibroblast, and plasma cells. So when it's time to discuss about the PanNuke dataset, this dataset is designed for nuclear segmentation and it has 205,000 label nuclei. This dataset will have 19 tissue types and it is having 5 cell types like neoplastic, inflammatory, epithelial, soft tissue, and dead cells. And these actually the models, the 5 different types of models which have said instance segmentation models which have chosen for this survey are tested on these 2 different types of datasets. This helped us to see how they perform. I mean, across different types of cells and also the tissues, which gives us a realistic view of their capabilities. Now, we use two types of segmentation approaches with the models, like instance segmentation, I mean, binary instance segmentation and multi-class segmentation. In binary instance segmentation, as we know that it will focus on only one cell type. Multi-class is like nothing but it will segments and labels multiple cell types in only one image. So with nucleus, we primarily focused on multi-class segmentation in this research because it is having, I mean, I just wanted to focus only on multi-class segmentation with nucleus. And as I have previously said, that it is having 13 cell types, while PanNuke, I performed both binary and multi-class. This PanNuke is having 19 tissue types. Next. So yeah, this is the overview of the PanNuke dataset and models which we have used in this study. So PanNuke dataset is having 19 tissue types and which is having 5 cell types. This PanNuke dataset is undergone with this models, mass CNN, Detectron 2, YOLO V8 and HoverNet. Next. Yeah, this is the training process. Like these are the steps which are common for every procedure. So data preparation, model configuration, training setup, model training, validation and evaluation, and at last post processing. I'm going to discuss about the evaluation step because in evaluation we have used different evaluation metrics. I'm going to discuss about them. So these are the evaluation metrics which I have used for my research: accuracy, precision, recall, F1 score, and IOU. Along with this, I also used AP and MAP, average precision, mean average precision also. So these metrics provided a comprehensive view of each model's strengths and also weakness. Now, yeah, this is the visualization of a single class instance segmentation, which is performed on only with PanNuke dataset. Like in this, I have used mass Gaussian, Detectron 2, YOLO V8 and YOLO V9, but it is only with PanNuke dataset. And the evaluation metrics which I have used are AP and MAP. I mean average precision, mean average precision. Now this is like multi-class instance segmentation, but with both nucleus and PanNuke dataset. So here I have used a mass Gaussian, Detectron 2, YOLO V8, YOLO V9, and also the HoverNet. With the different types of evaluation metrics, you can have a look of this now. So finally, in multi-class, particularly in PanNuke dataset, multi-class segmentation, HoverNet performed the best across all the metrics. So you can see here, multi-class segmentation, I have highlighted HoverNet performed the best across all the metrics. In binary segmentation, YOLO V8 shows the strongest results, especially for neoplastic cell. When I want to discuss about this nucleus dataset, YOLO V8 achieved the high precision, and mass CNN was effective for inflammatory cell type, while Detectron 2 was excelled with neoplastic cell types. So these results shows that each model has its strengths depending upon the task. So which makes it to choose the right model based on the specific needs and pathology. Yep. So you can see this visualization of results. These are like multi-class. So you can see that these are like multi-class instance segmentation, which are performed with nucleus dataset and PanNuke dataset. Here N1, N2, N3 are like, N4 are like with N1 represents mass Carcinogen, N2 represents Detectron 2, N3-YOLOV8, N4-YOLOV9. These are all related to nucleus dataset. Whereas P1, P2 are like P1 represents mass Gaussian and P2 represents HoverNet, which are related to PanNuke dataset. Yep. And next, I'm going to show you the visualization of a PanNuke dataset, which are performed only with which are mainly related to single instance segmentation. Here, mass Gaussian and detector onto YOLOV8 and YOLOV9 models are explored. So you can see that these are the regions which are like detected one, and this is the original mask of the image, the original image. So these are the single classes, class types that are segmented. So AG is nothing but the cell type, tissue type. C is nothing but the cell type. So in adrenal gland, connective cell type, this is the mask, original mask, and these are the predicted mask. Here BD is nothing but AG, BD, BR, LF, LBR. These are the labeled tissue types. C, N, I, E are like cell types. And then, so this is all about the explanation of single instance segmentation. To conclude, these research shows that importance of choosing the right model and applying the effective pre-processing techniques. So our findings can help researchers in selecting the best model for specific diagnostic task. So, as a GRA, Graduate Research Assistant, for my professor Dr. Sanghoon Lee, I am truly grateful for his guidance and suggestions. I would also like to thank Department of CCSC for hosting this C-Day. Thank you for your time.